So my name is Brian Browning. I'm at the University of Washington. Work a lot in methods that use haplotypes, especially for large-scale analyses, phasing, imputation, IBD segment detection, and others. So this is supposed to be a tutorial talk on identity by descent. So an identity by descent segment is just a segment of DNA that two individuals have inherited from a common ancestor, typically a recent common ancestor. In general, because it's inherited from an ancestor, you expect the two individuals to carry the same sequence of alleles. In general. I mean, there can be mutation and gene conversion since the common ancestor, but that's the exception. Most alleles will be the same, and that's the most straightforward way of trying to identify identity by descent segments is the long sequence of alleles shared. You might wonder, what about recombination? Can an IBD segment contain crossover recombination? The answer is no, not crossover recombination since the common ancestor, because that's what ends an IBD segment. Can't contain it because that's when you come to a crossover, you're starting a new segment. And that can be seen in this Wikipedia picture where there's 3 generations. In the top generation, they code each of the founders in the pedigree. Each haplotype is given a different color, which shows up, it's a little hard to see on this screen. In each generation, you can see crossover recombination occurring. So if you just look at this individual right here, you can see that from the father, they inherit the red haplotype, and then there's a crossover recombination, it changes to a blue haplotype. For the second haplotype, from the mother, you start out inheriting a green or olive. I guess in this picture it looks green to me, green haplotype, and then it switches over to a purple haplotype. So you get this recombination that occurs, and they point out that here in the 3rd generation, there's this orange segment that the two individuals in the 2nd and 3rd haplotypes here, they've inherited from the founder. That would be an identity by descent segment, a segment inherited shared and inherited from a common ancestor. And the segment ends when in one of the haplotypes the color changes. And why does the color change? Well, because there's been a recombination since the common ancestor. So when that recombination event occurs, all of a sudden the DNA material being transmitted down, instead of coming from one parent as it has been, now it switches over to coming from the other parent, or from the other haplotype, I should say. And so now you have a new common ancestor. You're starting a new IBD segment where you still have a common ancestor, but it's some other distant common ancestor. So because IBD is so tied to recombination, it's typically measured in a unit that's defined in terms of recombination called a Morgan. So a Morgan the segment length is X Morgans if X crossover recombinations are expected in the segment per meiosis. And it turns out the Morgan is so long it's sort of unwieldy. So we scale it down and typically use a centimorgan, which is just 1/100th of a Morgan. And on average, a centimorgan is around 1 megabase, but that's just an average. It can be more, can be less, but it gives you an idea of scale. So when we talk about a centimorgan, if you think, well, it's around a megabase, you're not too far off. And the length of IBD segments that we can detect accurately in outbred populations, we can get about down to 2 centimorgans, but after that, the error rate starts going up. So 2 centimorgans is about the length we're looking at. So that's a pretty long stretch of DNA, 2 million base pairs on average. IBD by itself is just a tool often for further downstream analyses. It can be used to detect genetic associations. This can be particularly valuable if you have individuals that don't know they're related, but you suspect they are, and they carry a very rare monogenic disease. So I saw an example of this where somebody came with data from a Native American tribe, where 2 people from the same tribe had this very rare condition. They didn't know they were related, but you can use the genotype data to find where they have IBD segments, and you can actually locate where the causal variant is. It can help you identify what variant is causing this rare severe condition. You can use it to detect natural selection. I'll give an example of that later. It can be used to estimate various quantities, relatedness, historical population size, migration, what's happened in the last several hundred years. You can use IBD in the current generation to give you a window of what's been happening to the population size, what's been happening in terms of migration, and estimating fundamental quantities that are important for understanding genetics, mutation, recombination, and gene conversion rates. So for detecting IBD segments, you're looking for long segments of shared alleles. That's the simplest way, the most straightforward way to do it. So I'll use this plot a lot where we have a long, this represents a segment of the genome, and then layered on top of that, there's implicit in all this, there's two haplotypes we're comparing. And in this segment, we just place where the discordant alleles are. And you can see in this big center segment that we have this long region with very few allele discordances, so this is a good candidate for containing a long identity by descent segment. I'll just put a green box around it, right? Because you can see compared to outside in the flanking regions, the rate of allele discordances is very low in this region. And if we could, of course I'm making this up what the IBD is, but the IBD might look something like this, where the color changes indicates that we're going from one IBD segment to a different IBD segment. And you'll notice that there's relatively few discordances per IBD segment. So the reason for that is you expect approximately one allele discordance due to mutation since the common ancestor per IBD segment. And why is that? Well, the rate at which per meiosis recombination occurs per base pair is very similar to the rate at which mutation occurs per base pair. So that equality of rates essentially leads to an average of about one discordance due to mutation per IBD segment. So since most IBD segments are very short, you get this sort of background rate of discordances. In most outbred populations, you have a discordance on average every 1 or 2 kb. That's just an average, but so fairly close together. But then you may have a long IBD segment that's 2 million base pairs with just one allele discordance. So the idea is very straightforward, very simple. And in the early days of my research career, when we'd have an idea like this, we'd say, my collaborators, Sharon, my spouse and my main collaborator, we'd say to each other, this is going to be an easy paper. And then after a few tries after that, it was said sarcastically, because there's no easy papers. Something always goes wrong. Matter of fact, we just say, it's going to be an easy paper, and the other person would just probably, what could possibly go wrong? It's like, yeah, right. All right, so the main part of this talk is talking about what could possibly go wrong. What are the things with detection methods you have to be aware of, design your methods to deal with, and also in downstream analysis be aware of. So there's 4 things that I came to my mind when I think of this, and the first is genotype error. So I've been giving you the nice clean case where all we have is mutation. But in reality, there's genotype error, and genotype error is independent of IBD status. So while you expect just one allele, discordant allele per IBD segment, the genotype error, I've added genotype error here, or at least my picture in my mind of what it's going to look like. And you can still see that there's a reduced allele discordance rate, but it's a lot less convincing here than it is there, isn't it? I mean, it just makes things hard, it adds a lot more uncertainty. So a lot of effort,
So instead we expect one mutation per IBD segment. Is that regardless of whether it's long or short?
All right.
Because the two are independent, right?
Well, all right, so if it's short segments tend to have an ancestor farther in more generations back. So there's more generations, more meioses for the accumulation to develop, right? So what are the strategies for dealing with this particular issue? Well, often several strategies, because this can be a severe headache when you're doing IBD segment detection. So you can apply strict marker QC filters. That's sort of probably the first one you think of. If it looks like this marker has a higher genotype error rate from our QC metrics, throw it out. If we're dealing with sequence data, we got lots of markers, we can afford to throw some out. One that's a little less obvious, but in practice we found it really helpful, is you apply a frequency filter. So in sequence data, most variants are very rare. Throw them out. You might be throwing out 90% of your markers. That's okay. They're not very informative, and due to LD, you aren't losing much power, and you're getting rid of a lot of potential headaches from allele discordances. So it's actually, in our experience, it's often a good trade to apply even a 5, 10% allele frequency filter. I mean, something pretty severe, throwing out variants with a lower frequency, and then performing your IBD segment detection. You can detect IBD with different marker subsets and merge the results. So the rapid method is sort of pioneered this approach. So in their approach, they take random subsets, and the thought is that, well, one of our subsets of markers, we can avoid one of them likely will not contain any discordant alleles, because discordant alleles are there, but if we take enough random subsets, we'll find one that doesn't have the discordant alleles, and that'll allow us to recognize that we have a long shared allele segment. So that's, often with almost everything we do, there's a false negative, false positive trade-off. So this is good for lowering your false negative rate for detecting things, but now you have a multiple testing issue. So to control your false positive rate, you might need to require a little bit more stringent thresholds, just like with multiple testing for association testing. You have to lower your p-value threshold to account for multiple testing. Here you might have to increase your length threshold. It's like, before I say something's IBD, it has to be a little bit longer to account for the multiple testing. You can use the seed and extend algorithm. Very simple, it's not probabilistic, but if you have a long shared segment, you hit a discordance. If there's enough shared alleles on the other side, you just jump across the discordance and keep going. So you seed it, and if there's enough shared, a long enough allele segment on the other side of the discordance, you just keep going. Seed and extend. In practice, this can work reasonably well, but it has to be tuned, of course, what parameters, what lengths things have to be. And what I like best is the probabilistic model that incorporates genotype error, but this also has drawbacks. Almost, everything from one has some drawbacks. Often the drawbacks, the trade-offs are worth it, but there's always drawbacks in methods. So here, one of the chief drawbacks is that it's a lot faster to compute a distance than it is to perform a probability calculation. So if you're using a probabilistic model, you are not going to be able to typically analyze as large a data sets as you can with models that just look at allele segment length. So the second issue is IBD segment endpoints. So here, going back to this picture we've used before, I've drawn the blue box maximally where I go all the way up to where it looks like we start getting a high density of allele discordances. So all the way here to this side. And you can see by drawing it maximally, I've got false positive IBD. I include a small blue segment that doesn't have any discordances, a little bit of this orange one, a little bit of the blue one at the side. See these endpoints, that's actually where all the action is in IBD segment detection, right? Every point in the genome is IBD. If you ask, is this point, do you and I share an allele IBD at this point? The answer is yes. Every point's IBD. Really the question is where are the boundaries, where you switch from one segment to another. And the major error mode is where you get the boundaries wrong. We're pretty good at detecting when there's likely a long segment, but then, we mess up on the boundaries. We either overestimate them or underestimate them and either get false positive or false negative IBD. In many of the analysis that I've been involved with, the false positive is a much greater concern than false negative. So we control that with a very unelegant but effective, simple but effective method. You just trim the ends. You take the ends, you push them in. Now, there's a trade-off. Now you're missing some of the green segment. But we've also got rid of the false positives. So if you're in a situation where the false positives are deadly, you trim. And in the analysis I've been a part of with outbred populations, we've been able to, when we needed to control false positives, a half a centimorgan trim, which on average is maybe 500,000 bases, half a megabase, seems to work pretty well. But there are cases where in-trimming doesn't save you. This is a case that until you stumble upon it or hear about it in a talk like this, you could easily step on this landmine. I have, right, until we stumbled on it. So you have regions of the genome where you don't have any marker coverage, right? It's highly repetitive, the reads don't map, you just don't have any data. So imagine one of these regions with a large gap where now you have the markers, you start getting genotype markers again, and by chance, they're concordant. These purple discordances don't occur immediately. You're doing IBD segment detection looking for long regions with identical alleles. It looks like, wow, this is it. I found it. But in reality, there's very little data under it. And so true, maybe the longest IBD segment in the whole region is over here. You do in trimming, you can see that most of what you get is going to be false positive. And so length methods, there are parts of the genome where length methods, if you're not careful, and I'll confess, I mean, in some of my early analysis, I was not careful. I didn't realize this was going on. You get a situation like this. So here are 4 length-based methods. We're counting along the genome how many IBD segments cover a point. You expect the coverage to be relatively flat, that the amount of IBD to be relatively flat across the genome. But here we got a spike, and notice the y-axis is on a log scale, so this isn't just a minor detail. This is a 100-fold, 1,000-fold, 10,000-fold spike in IBD. All of a sudden we're finding, 2 to 4 orders of magnitude more IBD in this central region, which, can you guess what's there? Centromere, right. Not all centromeres we come across had this spike, but here's an example of one. Yeah.
I have a question. So is one way that, okay, let's say that if you go to your previous schematic, you went and found, you have sequence data, and you went and found all possible callable sites. And it turns out that that chunk in the middle is monomorphic. There's no repeats. Could you call that like a true IBD segment there? It's just when you go down to the BCF, right? You would have no.
Yeah, if you don't have the markers, that's where you're because you think it's like the rest of the genome, and you've got loads of data here supporting you. In fact, you have nothing.
Yeah.
Yeah. Yeah. So, if you do a plot like this, you can find these and you can make adjustments post hoc, and throw out parts of the IBD segments that overlap this, and if after you throw out parts that overlap the spike, throw out the rest. But, ad hoc methods, sometimes it's tough to know where to set the thresholds because we have other spikes that are on the order of magnitude in other places. What do you do? And like I said, trimming, when you get a big enough problem like this, trimming isn't going to save you. All right, so here's where that probabilistic model comes in. So this is from UK Biobank data. So here's the IBD rate. So before we were counting the number of segments crossing a point. This is a scaled version of that. It's the proportion of haplotype pairs that are IBD at a point. So it's just think of what we had before, but scaled. But now there are many fewer peaks. And if you look at the scale, we no longer on a log scale. So the peaks are much, much smaller. And the most beautiful part is the peaks that remain actually have biological significance. Before, we had so much noise you couldn't see anything. I mean, the noise drowned out the true signal, but now the noise is gone. Look at the peaks. LCT, lactase persistence, under selection. MHC, major histocompatibility complex, under selection. OAS, you go to literature, you find this has an introgressed Neanderthal allele, under selection. TRPM1, pigmentation gene, under selection. So it got rid of the noise, and what's left, this just shows 4 of our top peaks, or 4 of the peaks from Sharon's analysis. I think we listed the top 12, and 11 of them were present in the literature. So we sort of had independent validation. There was one that we think's real, but it wasn't in the literature. So probabilistic methods for modeling, for handling marker deserts, work very well. Essentially what you're doing, this method, IBDN's method does, is at each point in the genome it builds a model for how long shared allele sequences are in that part of the genome, so it can recognize when something's unusually long. It also builds in a model for IBD, yeah.
IBDs were like segment lengths. How did you get from that to IBD rate per position?
IBD rate per position? So what we're doing is we take a position of the genome, we did our pairwise detection, we look how many segments overlap that position. And so the IBD rate is the proportion of haplotypes that are pairs that are in a segment at that point divided by the total possible number of haplotype pairs.
But do you threshold on length? So here you're looking only.
There was a threshold on length, yeah. There's a threshold on length. Based on what we've done before, I don't remember the details, but it'd be typically 2 centimorgans. But I'd have to go to the paper to confirm that.
So recent, really recent IBDs.
Yeah, if they get too small, the error, the ambiguity in the endpoints is very large relative to the length of the segment.
Yeah.
So 2 centimorgans is typically about as low as we go on length.
How many like generations ago would that correspond to?
So 2 centimorgans would be 50 generations connecting two individuals, so 25 generations back to the common ancestor, roughly, just back of the envelope calculation. So you're talking 600-700 years in that ballpark. If it's 2 centimorgan, if it's longer, the common ancestor could be shorter. All right, fourth issue, quadratic scaling. This is a different flavor, but we're in a computational institute. You got to talk about computation. Just starting out with just a plot from algebra class, right? Y equals x versus y equals x squared. Because of this difference in scales, this y equals x looks like it's flat. The whole point here is just to, remind people that when you have a quadratic scaling function with that, which means if your algorithm, you double the input size, the compute time goes up by a factor of 4, not 2. So it goes up 2 squared or quadratically. When it's quadratic, you quickly, the compute time grows so fast that you can't handle the large sample sizes. So this is, unfortunately, pairwise IBD segment detection is intrinsically quadratic. Why? Well, you double the number of samples, the number of sample pairs or haplotype pairs goes up by a factor of 4, right? So just a certain proportion of those pairs will be IBD, and so doubling the sample size, you expect the IBD output to go up by a factor of 4 in size. So asymptotically, IBD pairwise detection is intrinsically quadratic. And that's sort of an unavoidable feature. What can we do to make it better? Well, we can't solve the problem, but we can at least alleviate the pain. So if we can make as much of our algorithm as possible linear and limit the part that's quadratic, we still have a quadratic algorithm. But it grows a lot more slowly, like this blue line. So it's just trying to make the best of the situation we can. And so in the context of IBD, I think the clearest example of that type of approach would be the positional Burrows-Wheeler transform from Richard Durbin. It's one of the references. The references I gave are sort of selected to illustrate papers that introduce sort of new ideas into the field, new approaches at a certain level. So for PBW2, I included Durbin's paper. It scales linearly in the number of samples. That's exactly what we want. And it sorts haplotypes at each marker. So here's a picture from Durbin's paper. And it essentially marches down. And at this point, the PBWT algorithm is at this marker. And looking backwards, it's sorting haplotypes in dictionary order. So each row here is a haplotype. And just like A is before B in the dictionary, if you look at this last column, 0 is before 1. And where we have zeros, if you go to the second to next column, you'll have zeros before ones. It's just like a dictionary. So just as in a dictionary, words that share the same long prefix are going to be next to each other or very close proximity. Words, haplotypes that share the long allele sequence reading backwards will be close to each other. So this is a big help for IBD segment detection. You no longer have to look at every pair of haplotypes. You can just look at haplotypes that are close to each other in the PBWC sorting. It just really restricts your search space. And so using an approach like this, method, rapid and HapIBD and other methods that based on this or similar type of approaches can scale up to 500,000 samples. But it gets expensive and it gets, I think you're sort of reaching the limit with existing computing technology with a pairwise approach. But is there a way to get linear scaling? Well, yes, but you have to change your point of view. So if you think in terms of IBD clusters, which is a different point of view I'll talk about a little bit, instead of IBD segments, what we do is at each point in the genome, instead of thinking in terms of segments per se, at each point, if two haplotypes share a long segment that covers the point, we put them in the same set, in the same cluster. So now, when you do output for each haplotype, instead of listing the IBD segments it's a part of, you say, this is in the first cluster, in cluster number 1. You index them, give them each cluster a number. And so now you can, in the output data, you can look at every haplotype that's in cluster 1 and you say, oh, those haplotypes are all identity by descent at that locus. And there's only one output data per haplotype, so it scales linearly, right? And you don't need a lot of IBD segments per haplotype. If haplotypes A and B are IBD, B and C are IBD, that means they have a long segment covering a point. B and C have a long segment covering a point. C and D are a long segment covering a point. That's enough to put A, B, C, D all in the same cluster, right? So you're not having to look at every pair of haplotypes from A, B, C, D. Just a few IBD segments are enough to construct your cluster. All right, so one of the themes of this talk is there's always trade-offs. So what could possibly go wrong? All right, well, what if IBD segments are inconsistent? So, you know that haplotypes A and B have a long IBD segment, B and C have a long IBD segment that cover a point. So you put A, B, C in the same cluster and you go on, continue your algorithm. But what if you put A and C in the same cluster? What if they do not have a long IBD segment? And you can think of two cases here, the case where everything is fine and the case where everything is not fine. Here's the good case. Well, A and B might have this black segment here. IBD, B and C might have this one. And in this region where they overlap, then you will put haplotype C in the same cluster. And that's okay because this will be IBD by essentially transitivity of IBD. If haplotype A is IBD with B here and B is C here, then A and C will be. It's just like transitivity of equality. That's fine. The problem comes when you've made a mistake in your initial IBD segments. So we detected, let's say, all the way for the black and the gray section, but what if this gray, we've overestimated the endpoints? And here, what if we've overestimated the endpoints here? Now the black IBD segments no longer overlap, but because we think we found an IBD segment that extends to here, we think we found an IBD segment that extends to here, we declare all this IBD and we're totally wrong, right? So, the picture that comes to my mind is the snowball rolling down the hill in the cartoons. It picks up snow. You can have cascading errors. You're putting things together that don't belong in the same cluster, and your error rate just explodes. So if you're going to do this approach, you have to have very tight control of your false positive rate. You have to try to minimize and have very, very low error rates for this kind of behavior happening. You can't get it wrong. You can't afford to get it wrong. So we developed a method that was able to control the error rates well enough. This is version 0.2 of IBD cluster. And essentially it uses a couple of the techniques we talked about. It uses the IBD ends algorithm, which is fairly accurate for getting an estimate of where the endpoints are, but it's not accurate enough. So then we take the sledgehammer and hit it with a trim. Take the trim in. And then once we've got these segments that shouldn't contain any extra IBD, we do the clustering, and then it seems to work well. So I want to finish with just an application, because this is showing you how to IBD cluster, but what do you do with it? And some methods, unfortunately, I wish everything would work well with this kind of clustering because it scales linearly, but some won't. But some actually work better with this. So I want to look at estimating gene conversion rates. So a gene conversion is when you pass down the parent and child, the blue haplotype gets passed down, but a section of the red haplotype gets copied onto before it's passed down. And that section, they tend to be very short, let's say, a few hundred kilobases on average, very short. So the first thing is, we know that the average heterozygosity rate in humans is like, you have a heterozygous site every 1 or 2 kb. In many cases, you won't even see the gene conversion. You can't detect it happen because there's no heterozygous sites here. So this region that gets copied over, it's identical to what it's overwriting. So you can't tell it happened. Your only hope to tell it happened is if there happens to be in that copied region a heterozygous site. So you have a different allele copied in, and then when you look at the child, you can see, oh, okay, I've got this blue haplotype, but I got this allele that doesn't match other blue haplotypes. So we can use this to try to figure out, what rate at a site do we have converted alleles. So we look at IBD clusters, and if that IBD cluster at a point, they should all be IBD. If they're carrying different alleles, multiple alleles, and in particular multiple copies of multiple alleles, then that we can use that to measure gene conversion rates, because you have to do some filtering. You have to filter out the possibility that this is a genotype error. And you can do a pretty good job of that by requiring multiple haplotypes in your IBD cluster to carry each allele, because that indicates this allele is being inherited, right? Genotype errors don't get inherited. And you can control for mutation by putting a minor allele filter on, because mutations since the common ancestor are so recent, they haven't had time to rise to high frequency, they're rare. So you apply minor allele frequency, that takes care of the case where this could be a mutation, and you're left with gene conversions. So we did this type of analysis, actually Sharon, this is Sharon's method. I was responsible for the computational side to get something that runs fast, but I mean, this is her work. So we took two data sets, TopMed and UK Biobank genomes, one's 38,000, one's 125,000. We applied filters to get rid of the mutations and genotype error, so we're pretty confident we're detecting actual gene conversions that have occurred since the common ancestor. And then we estimate gene conversion rate as the fraction of IBD clusters that have discordant alleles after this filtering. So it's a relative rate. You can see at each point of the genome. And then Sharon did an analysis looking at replicability. She took sites that had the highest gene conversion rate in the TopMed data and looked at what are they in the UK Biobank data. And when she showed me the results, it's like, wow. It was like, this is great. All right, so this dashed line in the center, that's where the hotspot, we're taking the 6 highest gene conversion hotspots in the TopMed data. The black dots here are the gene conversion rates estimated in the UK Biobank data. And did it replicate? Yeah, I mean, things don't always work out this nice, but it's really nice when they do. In every case, we have this massive gene conversion hotspot related to the background. And then it's just sort of an extra, the cherry on the sundae, the resolution's so good, you can actually see the width of the gene conversion segment. These markers are filtered a little bit for various reasons, part of the analysis, but you can actually see where the segment starts rising up here, and it's particularly strong in this one. So you can actually study how wide the gene conversion rate is, the hotspot is, and it gives you an excellent way if you want to start investigating factors, some of which we know, which affect gene conversion hotspots. You can find them and determine the magnitude and width and so forth with this type of approach. I thought it was just a beautiful plot. These x-axes are all on the tick marks are 2 kb apart, so it's like having an electron microscope looking at this region of the genome. So thank you very much. Knowledge, any of this smart stuff was done by Sharon, and then funding and the data sources. Yeah.